Alrighty, good morning. We're going to talk a little bit about DNA, mitosis, and cytokinesis. So the DNA part is a little bit of a review. Um, so we're going to start out with a little bit of reviewing the background. There's a couple of people that I want to talk about. The first is that had to do with the discovery of the DNA molecule, specifically its structure. The first being Johann Friedrich Miescher, Miescher, about 1868. And what he did is he actually collected cells from pus and sperm. So really, really interesting stuff that he was working with. So he collected cells from pus and sperm, and from that studied the nucleus and what was found inside of the nucleus. At the time, he called it, the stuff inside of the nucleus. Obviously, today we know that he was looking at the DNA or the chromosomes. He figured out that what was inside of the nucleus was a slightly acidic substance, which makes sense since DNA is a nucleic acid. So makes sense that what he was looking at was slightly acidic. And he also noticed that there was a notable amount of phosphorus inside of those nuclei. So again, makes sense with the DNA having a phosphate group, so which it makes up that backbone, those sides of sugars and phosphates. So makes sense that he found it to be acidic and that there was a large amount of phosphorus. The next individual was Erwin Chargaff, and he did his primary work with the DNA structure in and about 1949. So he performed experiments and came up with two conclusions, which we know as Chargaff's rules for base pairing. The first being that, I guess he came to two conclusions, one of which was the Chargaff's rules for base pairing. I'm sorry. The first thing that he noted in his studies was that the amount he knew that there were he knew there were nitrogenous bases. So he knew about that there was adenine, thymine, guanine, and cytosine. From different species to another species, the amount of adenine, guanine, and thymine was different, so which implied that there were different amounts of DNA in various organisms. So one animal might have had 500 bases, and the other one might have had 5,000. So they all had adenine, guanine, and thymine, but what was different, it was the number of bases. So that was the first conclusion he came to. The second conclusion being his two rules for base pairing, is that he noticed, and it's just specific to a species, but if you were studying a group of species, the amount of adenine was equivalent to the amount of thymine, and the amount of cytosine was equivalent to the amount of guanine. So, and that's how he came up with his base pairs. Now, the third base pairing rule that we usually credit to Chargaff as being his third rule is that the number of purines equals the number of pyrimidines. So in the purine bases, if you recall, are adenine and guanine, and then the pyrimidine bases would be thymine and cytosine. So, so those were his two things, that if you were looking at different groups of species, the amount of bases would be different, but if you looked at the bases within one particular species, the amount of adenine would equal the amount of thymine, and the amount of guanine would equal the amount of cytosine. So the third set of people is Rosalind Franklin and Maurice Wilkins. They're notable because Rosalind Franklin is credited with, now she's credited with having an instrumental role in determining the structure and shape of DNA. Where Maurice Wilkins comes into play, she was, at the time, she was working in his laboratories. So to determine kind of what the structure of DNA was. So she was using his laboratories. Specifically, she was using X-ray crystallography or X-ray diffraction. So to try to see if she could determine what the structure was, and that's where that photo 51 comes from. And so with her study, she was able to deduce the following things. DNA had to be long and thin. It wasn't going to be super compact in its natural state. Now, when we talk later into the cytokinesis and mitosis part, the DNA does get compact, but in its resting state, it had to be long and thin. She also figured out that it had a approximate uniform diameter. So the diameter was uniform all the way through it. It was approximately 2 nanometers in diameter. The structure itself was repetitive, so it was made up of smaller repeating subunits. She didn't necessarily know what those subunits were, but she could tell that they were repetitive. And she wasn't able to distinctly say, but her famous photo 51 kind of led to the idea that it was helical in shape. Think of it a helical like a circular staircase. And then the last group of people are the most well known when it comes to the structure of DNA, and that would be James Watson and Francis Crick. 1953. They were a pair of chemists who used Franklin's research and some models of their own to kind of devise the actual final structure of DNA. They didn't actually do any research themselves. They just they kind of put things together. So they synthesized everyone else's ideas to come up with the final structure that we now know DNA to be. So they published their findings in the very well-known scientific journal called Nature, and which is where they talked about the structure of DNA. If you look in your packet, which I know there's a lot of extra pieces of paper this time, I actually included the article that was published with their in the journal Nature. So so yeah, I mean it's kind of cool to see that is that's kind of there. Linus Pauling, if you look on the second page, they do give some credit to other scientists. Linus Pauling would be number one if you want to look him up sometime. Kind of a well-known in the biology world. If you looked at number 3, Erwin Chargaff was credited, and as was number 6, if you look down there, Maurice Wilkins. However, Rosalind Franklin's name is left out. But it's just kind of interesting to kind of see the actual paper that they wrote. So that's just kind of for you. The next set of papers in your packet kind of show you that structure of DNA. You can kind of see the repeating nucleotides there. You can see the bonds. Specifically, adenine and guanine have a triple bond. You can kind of see that if you turn your paper sideways. You can see the different hydrogen bonds. And then adenine and thymine are going to have a double bond. You can see those phosphate groups off to the side. So, which is what Meister was able to figure out. If you flip, oh, never mind. The next part is going to be talking more into the mitosis, just the mitosis rather portion. So, all right, DNA structure. DNA stands for deoxyribonucleic acid. Deoxy meaning no oxygen, ribo referencing the sugar, ribose, nucleic acid talking about which macromolecule it was, or it is rather. So it is a polymer of the nucleic acid. So the macromolecule nucleic acids. So the other two major nucleic acids are RNA and ATP, or adenosine triphosphate, which is the energy. It was Meischer and kind of a little bit of Franklin also. They both were able to figure out that the structure was repetitive. So they, if you recall, that smaller piece is called the nucleotide. And the nucleotide, remember, is made up of a sugar, a nitrogenous base, and a phosphate. And those nitrogenous bases are the adenine, thymine, cytosine, and guanine. So that's just kind of the basic review of DNA. So this is important because when we go through the cell cycle, there is a part where the DNA has to be replicated. So we'll talk briefly about that, but this was just kind of a little repeat history about DNA. So we'll move on to the cell cycle. So there are three major phases, I guess four major phases, I guess. The last one, cytokinesis, is it's not really a phase, and it's just kind of a step. I'm not really sure what you would term that as, but three major parts. You've got interphase, mitosis, and cytokinesis. Interphase is then broken down into three subphases, although we still call them phases. So interphase is going to be composed of the G1, the S phase, and the G2 phase. So interphase is the longest of the phases. So the cell is going to spend most of its life in interphase, and then it will go through mitosis, or nuclear division, where the nucleus is going to divide. So you end up with two identical nuclei. And then in cytokinesis, the cytoplasm splits, the cell membrane splits, cell walls split, and you end up then with two separate cells. And then it starts the process all over again. If you look at your diagrams that I drew you on the front side, I've drawn a pie chart there. It's not exact in terms of what percentage of the cell spends its time, other than it does spend the majority of its time in interphase. Mitosis is a smaller amount of time, and cytokinesis is even smaller. So it's not exact, so don't quote me, because right now it looks like your S and G phase look like they make up 50%. I don't know that that's exactly the right percentages, but you get the idea there. So yeah, but we'll start with interphase and going into those particular steps. So the very first phase inside of interphase is called the G1, or the gap phase. This is where your cell is going to start growing, and that's really all it's going to do. It's just going to grow in size because it just got done dividing. So it's quite small at this point. So it's going to grow in size. The next step is the S phase, also known as the synthesis phase. I guess I didn't write that next to it, but the S stands for synthesis. I guess I did on the diagrams that I drew you, but I didn't on these notes here. Synthesis meaning that we're going to synthesize DNA. So this is where your DNA replication comes in. So I know we're flipping back and forth through a lot of stuff, but if you go back, if you flip through to your packets, further into your packet, the next page that after the DNA structure has a picture of the cell cycle, which I'll come back to here in just a second, but flip it over to the next one, and it's going to show you the process. I probably should have flipped these, but this was the order my professor had them in. but it probably would make more sense to move this. It's page 12 if you look down in the corner. But this is showing you how your DNA is going to replicate through semi-conservative replication. And if you remember, semi-conservative just means that half of the original molecule stays with it. So you can see in the diagram here that it splits, and then you end up with two identical copies of DNA, and the outside strands are the original, and then the inside two strands on the inside of that bottom there in figure 13.5, 15, sorry, 13, figure 3.15, sorry. The two inside strands there, those are the new ones. So that's why it's called semi-conservative, because you're going to conserve part of the original strand. So, okay. So that's what happens in the DNA replication. At that point, your chromatin, your piece of DNA, your pieces of DNA will then become chromosomes. And a chromosome, the definition of a chromosome is essentially that it's two identical chromatids that are going to be connected by a centromere. You might be thinking, what? So if you look at my doodle that I drew you, it's the bottom part of the page underneath the cell cycle image, the circle. Up on top, I've got what's called an unreplicated or an unduplicated chromosome. It's my very bad drawing of DNA, and you can kind of see that that big dark spot there in the middle. That's a centromere, which is just a bunch of proteins that normally hold two chromatids together, but not always. And so I've kind of drawn you the little circle, the two little circles around that to make it kind of a simplified version, so you wouldn't have to draw the actual chromosomes there. But that top one is a one chromatid chromosome. So this is what DNA is like before it goes through the S phase, before it gets replicated. After it gets replicated, if you look below that picture, it becomes that duplicated chromosome. So you've got two identical chromatids. Okay. Now the misconception is it's your chromosome is not while it looks like an X shape, the two chromatids are actually side by side. So if you look at my little picture of the duplicated chromosome on the right, I've done a better job of identifying that. So the left side of the chromosome and the right side of the chromosome, those are identical. It's not divided, top and bottom, or it doesn't go through as the actual X, where the one piece of chromatid would be like the top left and the bottom right would be one piece. That's not actually how they're held together. So I hope the diagram has shown you that. If it doesn't, you can just ask me and I can show you kind of with my hands. I would show you now, but I've got the screen share on. So, but anyway, so that's kind of what's happening in the S phase. So we've now duplicated and we've got two copies of each chromatid. So we call those sister chromatids because they're identical. Ideally, they're identical. Hopefully, there's no mutations that have happened while the chromosomes while the DNA was replicating. That left doodle with the squiggles, that's kind of what it looks like, but with the DNA, but I've drawn it kind of simplified there on the right. So, but that X shape is the chromosome, and then the single one up above is going to be a chromatid. So, and then the centromere there in the middle of that right diagram, that's going to be important here during mitosis, because that's where spindle fibers are going to attach. So, okay. So, the next part is the GAP2 phase, which is the G2 phase. This is where your cell is going to start preparing to go through cell division. Okay, so your cell is going to start preparing and making microtubules, which will eventually become your spindle fibers and your centrioles and things like that. Excuse me, that your cell will need in order to be able to separate the chromosomes. So gap 1 is growth. S is making DNA replication. So DNA replication, copying your DNA. Gap 2 is where you're going to start getting ready for cell division. So okay. Sorry, we've got all the different pieces of paper, so I just want to make sure we're all good here. All right, so go ahead and flip your diagrams over, and I've drawn you like, what, a billion little boxes of to show you the steps of mitosis. So we'll start walking through this. I'm going to go back. So we're now leaving the interphase part of the cell cycle, which is where the cell spends most of its time, and we're now entering the mitosis part. Sometimes it's called the M phase. Sometimes you'll hear it called the M phase. If you look at page 11 of your packet, so after the diagrams, in that circle, they call it the M phase, but where you have mitosis and cytokinesis combined, but sometimes you see them separate. So mitosis is specifically called nuclear division because you're going to divide the nucleus. So the very first step in mitosis is prophase. You're going to go to prophase, metaphase, anaphase, telophase, or PMAT. That's how I remembered it. So prophase is the very first step. If you look at the diagrams that I've drawn, you've got my very pretty drawing here. I start with interphase. So you've got the cytoplasm. Your DNA is inside of your nucleus. You can see the nucleus. You can see the nucleolus. It's usually a little dark spot. And yeah, that's interphase. So once we leave interphase and we start prophase, then we're in mitosis. The very first thing that starts to happen once we enter the mitosis part of the cell cycle is you start seeing your chromosomes. They start to actually condense a little bit more. They become more compact. I don't know if that's what I just said or not. It's kind of late, so I'm not sure if I'm repeating myself. So if I am, I apologize. But you can definitely start seeing the chromosomes. If you look at the next one over, so on the top row, the one on the far right, I should have numbered these. So it would be the third picture. I've got on there that the chromosomes are fattening up a little bit, so getting more compact. You can see the centromeres, and remember, you could have the 2 sister chromatids per chromosome. Your nuclear envelope starts to dissolve, and your nucleolus starts to dissolve. That's what's happening in pictures 3 and 4. And this is all during prophase. So a lot of stuff happens in prophase. The last picture of prophase is picture number 5, I think. And this is where your microtubules that you made in the gap 2 phase, back in interphase, that's when they're going to start getting put together and forming spindle fibers, which are those long lines, I guess, coming out from the top and the bottom. So the dots there at the poles of the cell, like we like to call them the poles, like the north pole and the south pole of the cell. Those are the centrioles. Again, spindle fiber, microtubules, and some proteins in there. So you've got microtubules becoming your spindle fibers, and the chromosomes are going to start moving towards the center of the cell. They're not going to be at the center just yet, but they're going to get there. So, and that's prophase. Now, the 6th picture there is we're now leaving prophase. We're now in metaphase. Okay, there's not much that happens in metaphase. It's a pretty quick one. But spindle fibers attach to those centromeres, and I've tried to draw that as best I can. You can kind of see that they're supposed to be attaching to the centromeres, those dark circles there. And then the chromosomes are lining up at the middle or the equator of the cell, we call that, and I have labeled that. That equator of the cell is called the metaphase plate. But your chromosomes are going to line up on that metaphase plate or at the equator. And that's metaphase. It's pretty straightforward. They line up at the center. Anaphase is the next one, so that would be, I think, picture number 7. I really should have numbered these, but I kind of think of them like a rod and reel, like a fishing pole. So during anaphase, what happens is the spindle fibers kind of start pulling the chromatids apart, and they break, and you end up getting half a chromosome or one chromatid at each end of the cell. So they line up to the point where they're basically breaking them, and the sister chromatids are being separated. So so you still have the same genetic information on each one. So so they're pulling them across, and that's basically anaphase. Picture number 8, they're really getting pulled to the pole of the cell at that point, so they're finishing where they're supposed to be. The last one is telophase. Some people pronounce it telophase. I don't know that it's right or wrong either way. I was taught telophase, so that's how I say it. But you can kind of see that the nuclear envelope is starting to reappear. The chromosomes are starting to loosen up. They're becoming less fat or less tight. That's what picture number 9, I guess. Picture number 10, the nuclear envelope in that picture is established, and the nucleolus starts to reappear. So you can kind of see in picture number 10 there on that bottom row that you can see that we're definitely going to have 2 cells at that point. Now, we don't have the structures in between to make it actually separate yet, but we're really, really close. That is technically the end of mitosis. We are essentially done with mitosis at that point. We're now entering cytokinesis. So yeah. These last 3 pictures are cytokinesis. So I quit drawing the chromosomes at that point, or the chromatids rather, sorry. So I just put 4 chromatids or 4 CHR, but you get the picture. So now the first step in cytokinesis is that there's a row of microtubules that form along the center of that cell. And you can kind of see how I've drawn those as little dashes. The next thing that gets formed is you get these pectin-filled vesicles. Pectin is basically what gives Jello the ability to gel, if you will, to make it that jiggly solidness that is Jello, I guess. I don't really have any better way to explain it, but pectin is what allows it to solidify. So you've got these vesicles that are filled with pectin, and they insert themselves in between those microtubules, which then become, they start adding in pieces of endoplasmic reticulum. It's going to start building these pectin-filled vesicles from the center out. So they start from the middle and then they work their way out. The Golgi apparatus makes these pectin-filled vesicles, and then those pectin-filled vesicles will move to the center of the cell there and insert themselves in between those pieces of endoplasmic reticulum. So this is what's called, I didn't label it, but the pieces of endoplasmic reticulum with the pectin-filled vesicles are known as a cell plate. So the cell plate. So if you ever hear during cytokinesis the cell plate starts forming, that's what they mean. I guess I should go on. Sorry. I'm just talking and I forgot that there was a slide there. So, so yeah. So the very, very last picture there. Oh, sorry. By the way, the second to last picture. So that would be picture number 12. With the pectin-filled vesicles and everything. You can kind of see now how I've drawn an inside layer there. So we're starting to establish the cell wall, and we're going to start establishing the plasma membrane here in just a minute. But it's important to note that we're now getting the multiple layers. So, all right, the very last step of cytokinesis. Your vesicles are going to burst and are going to release the pectin. The pectin, that gel, is going to serve as the glue that's going to hold those pieces of endoplasmic reticulum together. And those are going to be left there because those are going to serve as channels that allow cells to communicate with each other. So that top cell will be able to communicate with the bottom cell through those little channels that have been left behind. And those channels are called plasmodesmata. And I've labeled that. So on your diagram. The little line that goes between the two is called the middle lamella. So your plasma membrane is also going to form. So now you can see that there's definitely two layers in these cells. There's the cell walls and the plasma membranes. So so yeah, that's basically how cells are going to divide and reproduce. At this point, everything would start back over. The cells would start entering an interphase again, and they would start getting bigger again and starting the process all over again. So a couple of the pages that I have in your packet are going to kind of show you better doodles of mitosis. So like page 13, for example, that top portion is showing you the duplicated sister chromosome, showing you kind of where the centromere is, showing you where the replicated chromosomes are. And then you've got the diagrams down below, which are a little better than my drawings of mitosis. If you flip the page again to page 14, I don't know if I'm on front or back at this point. I think I'm on the back side. This is going to better show you how the sister chromatids separate in mitosis. So even though I was drawing them going to the north and south ends of the pole, this diagram, it looks like they're showing them being separated east to west. But that's how, I mean, they would orient themselves to be essentially sideways if they're going to get pulled north to south, so that you can kind of see how they're getting pulled apart there in on page 14. So page 15, it's just kind of giving you how many chromosomes are in different types of plants. So this is kind of showing you that Chargaff was point that the number of bases was going to be different, so the number of chromosomes is going to be different also. So it's just interesting facts there. And then table 4.2 is showing you kind of about how long it takes for a cell in a pea root, how long it's going to take for it to grow. So kind of at room temperature and a little bit colder. So it kind of shows you the breakdown of how long interphase is, how long prophase, metaphase, anaphase, telophase is. So obviously you can tell by those numbers that interphase is definitely the longest. So anaphase is the shortest. So it splits pretty fast if you think about it. Looks like the entire process takes about an hour and a half to divide your nucleus. So the last page on the back, I think it's the last page. The last page in your packet, there's a little bit on cytokinesis there, a little bit more information on cytokinesis. And on the back side, kind of talking about the plasmodesmata. So kind of showing you some more diagrams, better doodles than I can draw. So some of the pictures are actual pictures from the microscopes, but they're kind of hard to see because the quality isn't so good. But okay, the last thing that I want to go over, I know this is a long one too, but the last thing that I want to go over is maybe, there we go, what you end up with at the end of cell division, specifically with mitosis. Specifically with mitosis, you are going to end up with two daughter cells. We start with a parent, what's called a parent cell, and then the daughter cell is left behind, and it is, you've got two daughter cells that are identical, hopefully. Hopefully there's been no, what's it called, no mutations to the point where one of them is drastically different. So now the chances of them being absolutely identical, not super great, just because it's possible that you're going to have one difference in a base here or there, but nothing substantial to make a huge difference. So, but ideally, at the end of cell division, at the end of mitosis, you end up with two daughter cells that are identical to one another, and they're also identical to the nucleus that they were derived from. So, so that's mitosis. Tomorrow we're going to hit meiosis, which is going to be really similar to mitosis, but it's you basically go through these 4 steps twice, and you end up with you end up with 4 cells instead of 2, because you end up going through division twice. So we'll kind of compare those two tomorrow. I know this was a lot, so if you do have any questions, please don't hesitate to ask. I am in the conference room, so I can't answer any questions. If you want to email me. And then tomorrow, the 7th graders will be taking a quiz during your hour. So if there's something that you want me to clarify a little bit, I should be able to talk to you tomorrow and clarify anything that you might need. So hope this makes sense, and that'll be it. So have a great day.